So welcome back to our study of psychology. We are moving into the biological basis of behavior, and today we're going to be going over what you see here as module number 9, biological psychology and neurotransmission. So going along with the same way we've been discussing things in the previous 8 modules, we're going to look at some of the targets of what we're going to be trying to understand today. The first one being able to explain why psychologists are concerned with human biology. I think sometimes some students, especially those students who come in with sort of an orientation of wanting to understand psychology from more of a clinical perspective and understanding disorders, they're like, whoa, this is kind of like biology and anatomy. Why do I have to understand this stuff? So we're going to be talking about that. You're going to have to be able to describe the parts of a neuron and explain how neural impulses are generated, understand nerve cells and how they communicate with other nerve cells, and describe how neurotransmitters influence behavior, and explain how drugs and other chemicals affect neurotransmission. So why are psychologists concerned with human biology? Well, let's think about this. You can't totally separate the mind from the body. Biological psychologists study the linkage and interplay between the body and the mind. So psychologists from this perspective study the links between our biology and our behavior and mental processes. So we should be thinking that everything that is psychological is simultaneously biological. And I like to think about it, and I think different textbooks explain it this way, and that's why I read it, that our nurture works on what nature provides. So over and over again in psychology, we're talking about that split between nature and nurture, and we need to be thinking about how much they both matter, and that nurture works on what nature provides. Historically, what has been really interesting is how a long time ago, Aristotle, who John Green always points out is wrong on a lot of things, he was also wrong on this. He thought that the mind was in the heart. Well, as we know, it's not in the heart, that the mind is actually in the brain. And the Greek physician Hippocrates is one of the first people to really correctly point this out. Even though we also associate love with our hearts, we do know that it is our brain that falls in love. That's where we find that connection with other people. So what is the basic part of our brain? Well, it's the neuron. It's a nerve cell that is the basic building block of the nervous system. So the mind and the body communicate through this electrochemical process. And that's going to be really important to understand, that it's both electrical and chemical. The process is going on inside of your brain, which is like crazy to think about when you start learning about this, and you're like, oh my gosh, this is going on with every single thought I'm having. As I'm sitting here talking, all these neurons are firing all over the place in my brain. And it is electrical because the neuron passes an electrical impulse along its axon, which we're going to talk about on the next slide. And it's chemical because at the ends of the neurons, the axon terminals secrete chemicals called neurotransmitters. So again, the neurons are the basic building blocks of the entire nervous system. And the best guess is that there are probably about 80 to 100 billion neurons in your nervous system, but that is a guess. Nobody's actually sat there and counted them, so that is a guess. What's important to understand is throughout life, new neurons are born and unused neurons wither away. Okay. And we're not going to get into it too much, but there are a few different types of neurons in terms of sensory versus motor versus inner neurons. There's some types, and but for now, we're going to talk about the basic parts of a neuron overall. And you'll see pictures like this over and over again. And there are some very basic parts of the neuron that are going to be really important to understand. So this is where psychology becomes very much like an anatomy class that you may have had. And this is just right now, I'm just going to go over an overview of this sort of the parts of the neuron and a little bit about the process of how neural impulses happen. So in terms of the parts of the neuron, you can see, let me see if I can get my cursor over here. Right here is the cell body, the cell's life support center. It has a nucleus in the middle. And then on the outside of the cell body, you have what are called these like feather-like fingers sticking out from the cell body that bring information into the cell. Okay, so those are bringing information into the cell. The dendrites are fibers that receive and integrate information, then conduct it towards the cell body. Okay. Then we have the axon. You can see the axon is this really long arm that sends information away from the cell body, and it's going to be passing it on to other neurons or other body parts. Okay. Axons are actually insulated by something really important called the myelin sheath. The myelin sheath is sort of this fatty material that covers the axons of most, I think, neurons and helps speed neural impulses. It's sort of like an insulator in that it helps the impulses kind of move more quickly. It helps the electrical charge move more efficiently. And what's really fascinating, and we're going to touch upon this again too, is that myelin is actually laid down on the axons of your neurons until about the age of 25. And what you're going to hear over and over again as we talk about the brain itself is that there's no magical time at 18 years old when you become an adult that your brain stops developing. I think there are actually quite a lot of things that aren't fully developed till about age 25 or even beyond. So the myelin being laid down until about age 25 is important. As this is developing, it's helping with neural efficiency, judgment, and it also seems to help be related to self-control. So what else? Do I talk about all the different parts here? Okay, so we've also got the terminal branches at the end of the axon that sort of form junctions with the other cells. So that the information, the dendrites are going to bring the information into the cell body, and then it's going to be pushed passed through the axon with the help of these, with the myelin sheath, and in between the myelin sheath, you've got these little spaces that are called the nodes of Ranvier, I guess named after someone named Ranvier, and they actually help the process along too, this electrical process, and that for the message to get moved from the dendrite through the neuron itself along the axon, all the way out to the terminal branches of the axon, and where it's going to connect in a synapse to another neuron or another body part. So just to get more specifically into those parts of the neuron that I just described, what is that cell body, or soma as it's called? It's the part of the neuron that contains the nucleus, the cell's life support center. Just like other cells in your body that have nucleus, the nucleus is the cell's life support center. The dendrites are the bushy branching extensions that receive, so remember the dendrites are receiving and integrating messages, and they're conducting those impulses toward the cell body. So there's like finger-like little structures are receiving information. They extend out from the cell body like branches and receive messages from other nerve cells. The axon is attached to the soma, the neuron extension that passes messages through its branches to other neurons or to muscles or glands. So the axon is a long single fiber, and they can be really long depending on like what part of the body they're in or what type of animal they're on. An axon is a long single fiber that translates messages from the cell body to the dendrites, from the cell body to the dendrites of other neurons, or to other body tissues such as muscles. Again, this is a really important component of what's covering most axons is myelin sheath. Myelin sheath is a protective covering that covers most neurons. It insulates the axon and helps those nerve cells become more efficient to travel faster and farther. It increases the transmission speed and provides insulation. So why is this concept of myelin sheath so important? It's important for many reasons. At birth, babies have not yet myelinated their axons of their neurons. The development of the myelin sheath is not complete until around age 25. Of course, there are variations in that, and it's crucial to many things like behavior, movement, and thought. Deterioration of the myelin sheath can lead to motor impairments, such as the disorder multiple sclerosis. And that's something you really want to point out, because I've seen that come up over and over again, especially if you're going to take an AP exam within practice questions and such, that there's often a question about, which disorder is related to a deterioration of the myelin sheath, and that would be multiple sclerosis. So here's a tip. If you are taking the AP exam, that questions about the myelin sheath often show up on the AP exam, as I just said. Often the question is about the purpose of the myelin sheath, in that it speeds neural transmissions, and other times the question is about the link between the deteriorating myelin sheath and disorders such as multiple sclerosis. Make sure you kind of understand that it's really important, because for most people taking this class, it's a really new term. You probably have heard about neurons, and you may have even heard about axons and dendrites, but the concept of myelin sheath may be something that you've never heard of. It's a new term, so you really want to wrap your brain around that term. It is important. So what are those terminal branches that were at the end of the axon? Well, the ends of the axon contain terminal buttons which hold synaptic vesicles that store neurotransmitters. Another really important type of cell are glial cells. They're cells that support, nourish, and protect the neurons. They also play a role in learning, thinking, and memory. If you think about neurons in your brain like the queen bees, they're the ones on their own. They can't feed or sheathe themselves, but they're the queen. They're the really important ones. Well, they're all really important. Glial cells are apparently really important too, just like worker bees. The glial cells are the worker bees. They provide the nutrients, insulate the myelin, they guide neural connections, and they mop up the ions and neurotransmitters. Glial actually means glue. These are the glue cells. These are so important within the functioning of to help function the neuron, to help the functioning of the neurons. So the concept of threshold is really important as we're thinking about this electrical, chemical information being passed throughout your brain. The concept of threshold is something that you really have to be thinking about within your study of the biology of the brain. What is a threshold? Well, it's a level of stimulation required to trigger a neural impulse. Neurotransmitters received by dendrites build up to initiate the action potential. And this is really important. It's like it has to build up to a certain point, and then it can fire, so to speak. So the thresholds are sort of the minimum amount of a neurotransmitter transmitter needed in the synapse to start a reaction. And we talk about this all or nothing response, the all or none principle. All action potentials fire at the same strength. More stimulation does not produce a more intense neural transmission. The neuron's reaction is an all or none response. Neural firing happens at full response or none at all. Just like guns, neurons either fire or they don't. Pulling the trigger harder won't make the bullet go faster. Well, I don't know of any guns like that. All action potentials are going to fire at the same strength. So neural impulses can be either excitatory or inhibitory. So an excitatory signal, think about it like the gas pedal of the car. Excitatory neurotransmitter signals trigger action. On the other hand, like the brake pedal of a car, inhibitory neurotransmitters signal depression of an action. So when excitatory impulses outnumber the inhibitory impulses, the threshold has been reached and an action potential occurs. Let's do a little bit of a learning check on some of the stuff because we just went over a ton of information in a really short amount of time. So in order for a neuron to generate an action potential, which of the following must be true? What did I just say? For a neuron to generate an action potential, which of the following must be true? So excitatory impulses must outnumber inhibitory impulses. C is the correct answer. What is the resting state of an axon? So we were just talking about when axons are going through the whole when neurons are going through the electrochemical process, but what is the resting state? Prior to the beginning action potential, the outside of an axon's membrane has mostly positively charged sodium, Na, think back to that periodic table, ions, and the interior contains negatively charged proteins and a small amount of positively charged potassium, K, ions. This creates a slightly negative charge, and at this point, the neuron is said to be in the resting state, or it's polarized. So when a neuron is not firing, it's in its resting state, called resting potential. When resting, the neuron is negatively charged. The outside of the axon is sort of surrounded by sodium. Inside of the axon is filled with potassium when resting. Some stimulus sends an arousal message, right? Something happens, like oh my, ouch, something like that. This stimulus causes your neurons to react. This is called an action potential or neural impulse. Another concept to understand is that the axon membrane is selectively permeable. I like that terminology, selectively permeable. I think I'm selectively permeable. I decide on what comes in and what comes out of my brain. The membrane contains voltage-gated ion channels that either open to allow ion exchange or depolarization in, or close to prevent ion exchange, polarization. So what are the steps in an action potential? Well, the first section of the semi-permeable axon opens its gates once the threshold is met. Sodium ions flood in through the channels. Why? Why did these sodium ions rush in? Well, since the inside of the membrane is slightly more negative, the sodium ions try to balance the charge. So think back to your chemistry classes. If you've taken some sort of chemistry, hopefully you have a basic understanding of that process. This causes slight depolarization. Then what is the next step in the action potential? Well, the depolarization changes the electrical charge of the next part of the axon. The gates in the second area are now open, allowing even more sodium ions to flow in. Then how do those potassium ions move out? At the same time, the gates open in the first part of the axon, allowing potassium ions to flow out. This repolarizes that section of the axon. Then the sodium-potassium pump continues to depolarize new sections of the axon and repolarize the previous sections. This is complicated, and you might want to go back and listen to this more than one time. How does the impulse move? The influx of the positive ions is the neural impulse. The impulse moves down the axon like dominoes falling one after another. The message in the form of an action potential continues on down the axon until the neuron fires or releases a message to the next neuron in the chain. The sending neuron's message is sent into the synapse, which is the little space in between the neurons. The message is a chemical called a neurotransmitter. The receiving neuron will take up the neurotransmitter, or the neurotransmitter can be blocked. In bad cases, in some cases, neurotransmitters can be sucked back up by the sending neuron. And this is where you hear the term reuptake. What happens after the action potential moves to the end of the neuron? Well, neurons need short breaks, just like all of us. During this resting period, it's called the refractory period, subsequent action potentials cannot occur until the axon returns to its resting state. Then the neuron is able to fire again. So again, after the neuron fires, it needs to chill out a little bit before it can fire again, just like anything. This is called that refractory period. The neuron is totally unable to fire again. After a brief refractory period, the neuron will repolarize. The sodium will go back outside the neuron, and potassium will return to the inside, and the charge will return to slightly negative. So let's review some of these key terms. Again, this is complicated. You may want to listen to this more than one time or read about it read over it a few times and quiz yourself on it to make sure you're really understanding these concepts. So polarization is the resting state of the neuron. The charge is more positive on the outside of the membrane and more negative inside. Depolarization, the action potential, the rushing in and out of positively charged ions. Repolarization is the refractory period, the closing of the membrane and reestablishing a more negative charge inside. So how do these neurons communicate with each other? The sending neuron releases neurotransmitters. You've probably heard that term. We hear about that a lot now in the popular culture, about neurotransmitters and these chemicals that are inside of our brains. So the sending neuron releases neurotransmitters across the synapse to the receiving neuron. Messages travel along a single neuron as electrical impulses, but the messages between neurons travel differently. The transfer of the information from neuron to neuron takes place through the release of these chemical substances. So remember when I said the communication within your brain is electrical and chemical, electrochemical. So the transfer of information from neuron to neuron is chemical in the form of these chemicals called neurotransmitters. So what happens is the neuron to neuron transmission takes place through the release of the chemical substances into the spaces between the axons and the dendrites of specific neurons and those synapses. The chemicals, again, are called neurotransmitters. And this process is called neurotransmission. Again, the spaces are called synapses between the neurons. When neurons communicate, an electrical impulse triggers the release of the neurotransmitter from the axon into the synapse. The neurotransmitters cross the synapse and bind to special molecules on the other side called receptors. Receptors are located on the dendrites. Receptors receive and process the message. And again, then it keeps happening over and over and over again. So what is a neurotransmitter? Well, it's a chemical messenger that travels across the synapse and binds to the receptor sites of the receiving neuron. A synapse, which I've mentioned several times, is the junction between the axon tip of the sending neuron and the dendrite or cell body of the receiving neuron. It's a tiny, tiny little gap. For a long time, I think scientists didn't even know they existed, right? And then I'm blanking at the moment, but someone discovered them somewhere in Europe, maybe Sweden or Switzerland. I can't remember. I don't think that's accurate. Someone discovered, okay, wait, there's a little bit of a time lapse here. There seems to be some sort of little space here in between these neurons, and they called it a synapse. This tiny gap at this junction is called the synaptic gap or synaptic cleft. So how does this process work? When an action potential reaches an axon's terminal branch, think back to one of those first pictures I showed you at the beginning of this session today, it stimulates the release of the neurotransmitter molecules. These molecules cross the synaptic gap and bind to receptor sites on the receiving neuron. This will either excite or inhibit a new action potential. So this concept of reuptake, you'll hear about over and over again. And when we get to clinical psychology, that topic, you'll hear about reuptake in the form of things like antidepressant medications and what they're doing within someone's brain to reduce depressive symptoms. So what is reuptake? It's a neurotransmitter's reabsorption by the sending neuron. Okay, so it would go back in. They were going to send it, but it came back in. It reuptook it. Sort of a strange word. So let's do a little comprehension check. What would you answer? When there is a negative charge inside an axon and a positive charge outside of it, the neuron is a negative charge on the inside and a positive charge outside of it. The neuron is said to have a resting potential. Neurotransmitters cross the what to carry information to the next neuron. We just talked about that like 2 slides ago. That is the synaptic gap. So now we're going to switch over to talking a little bit about neurotransmitters in general. And what are some of the ones that you want to be aware of within AP psychology? So here, I'm not going to read through every single thing on here, but I'm going to leave it up here for a few minutes. We're going to talk about a few of them. a few of the important neurotransmitters, like acetylcholine, which enables muscle action, learning, and memory. And you can see what it's associated with Alzheimer's disease. Dopamine is a really interesting one that seems to influence movement, learning, attention, and emotion. And having too much of it has been linked to schizophrenia on the one hand, but having too little of dopamine is linked to decreases in mobility and things like Parkinson's disease. Serotonin affects your mood. That's why when we get to the clinical psychology topic, you'll hear about serotonin, and it talks about depression and medications that are working on those serotonin receptor sites. Like it says right there, an undersupply is linked to depression, and some drugs that raise serotonin levels are used to treat depression. Norepinephrine helps us control our alertness. If you don't have enough, you can feel a depressed mood. GABA is an inhibitory transmitter, and undersupply is linked to seizures, tremors, and insomnia. Glutamate, this is one I'm recently very interested in because I've made the connection between having too much glutamate in the form of MSG. It's totally correlational, but I seem to notice that I do get really bad headaches when I have food that has MSG. And it does seem that it might not be just correlational. I may be onto something there, that it probably is causing me to have a headache. And it doesn't happen to everyone, but it seems to happen to quite a few people. And then endorphins are neurotransmitters that influence the perception of pain or pleasure. You've probably felt endorphins if you've ever had like a big rush from going on a run. I used to have that happen. I even ran a marathon once and thought it was fun, but I don't feel that anymore now that I'm older in terms of getting that endorphin rush, but hopefully some of you still do. You get that sort of runner's high, this pleasure that these endorphins are released into your body, and you feel really, really good. And that's sort of what opiate drugs do artificially. The problem is, of course, that you become very addicted, and then those opiate drugs actually suppress your body's natural endorphin supply, and you can no longer create that natural high. So neurotransmitters have different neural pathways. So this is like the serotonin pathway on the left versus the dopamine pathway on the right. So let's consider the neurotransmitter acetylcholine. It plays a role in learning and memory. It's a messenger between motor neurons and skeletal muscles. When it's released to our muscles, cell receptors, the muscle contracts. If its transmission is blocked, the muscles cannot contract and we're paralyzed. It's very, again, helpful with neuromuscular movement found in the synapses of motor neurons and skeletal muscles. And again, a decrease in it is linked to Alzheimer's disease. Let's consider dopamine. So you guys are probably familiar with this person who is a famous boxing legend who suffered from Parkinson's disease. And dopamine seems to play a large part in Parkinson's, and that there appears to be an undersupply of dopamine. Dopamine is released in response to feel-good behaviors and produces euphoria and pleasure. Many illegal drugs mimic dopamine. And again, ineffective dopamine receptors in the body are linked to Parkinson's, whereas too much dopamine in the brain is linked to schizophrenia. A little bit of the same thing I just said on the previous slide, just the importance of dopamine being associated with rewarding feelings and somehow related to having a role in addiction in general. It's believed that excess levels are, again, this is an important concept, excess levels of this neurotransmitters in the synapses is linked to schizophrenia. So what are endorphins? So look at the word itself, and you can see the parts of it and how it's like an endogenous morphine, meaning morphine within, natural opiate-like neurotransmitters linked to pain control and to pleasure. Again, that whole idea of runner's high. How do drugs alter neurotransmission? Well, drugs and chemicals from outside the body alter our brain chemistry. Drugs can act as agonists, exciting neuron firing, or as antagonists, inhibiting neuron firing. So how does an agonist work? Well, an agonist is a drug molecule that increases a neurotransmitter's action. So a neurotransmitter that would open the receptor site, agonist mimics the neurotransmitter opening it, okay? So if you have a certain type of agonist drug, it would mimic a neurotransmitter that was opening a receptor site. Agonist means it's exciting. It has an excitatory effect. It's saying go. It makes it more likely that the next neuron will fire. Drugs often mimic our neurotransmitters and trick the brain. It can also block the reuptake of a neurotransmitter. How about an antagonist? Well, an antagonist is a drug molecule that inhibits or blocks a neurotransmitter's action. Antagonists can also work by blocking reuptake. Antagonists inhibit. They're like, stop! They make it less likely that the next neuron will fire. Okay, this is a ton of information today. So we're going to do a little bit of a review of our learning targets from that second slide that we went over. That first learning target was explain why psychologists are concerned with human biology. Hopefully you have a little bit more of an understanding of that now. Researchers working from a biological perspective study the links between our biology and our behavior and mental processes. We are biopsychosocial systems. Biological, psychological, and sociocultural factors interact. If you remember from previous modules, that biopsychosocial triangle is so important as we go through AP psychology. These three systems are interacting to influence our behavior. Our second learning target, describe the parts of a neuron and explain how neural impulses are generated. This is complicated and very much like anatomy class. Neurons are nerve cells that consist of a cell body, dendrites, axons, some of which are encased in a myelin sheath, and terminal branches. Glial cells are other important types of cells that support nerve cells and produce that myelin that's so vital. If the combined excitatory signals exceed a minimum threshold in a neuron, the neuron will fire, transmitting an action potential. It's important to remember that the neuron's reaction is an all or none process. And I want to take some time you should practice and understand the parts of a neuron. It's really important to really be able to visualize that and understand how this process is going. So by, creating your own 3D model of a neuron, drawing a neuron, printing out a picture of a neuron and coloring in the different parts is an important and easy way to study and understand the parts of a neuron. And then thinking about how the process of that impulse happens. So describe how nerve cells, the next learning target, is describe how nerve cells communicate with other nerve cells. When action potentials reach the terminal branches, they stimulate the release of neurotransmitters, which then carry a message from the sending neuron across that synapse to the receptor sites of a receiving neuron. Reuptake, an important word, is the reabsorption of excess neurotransmitters. The last learning target was describe how neurotransmitters influence behavior and explain how drugs and other chemicals affect neural transmission. So neurotransmitters travel designated pathways in the brain. So different neurotransmitters travel in different ways in different places. Acetylcholine affects muscle action, learning, and memory. Endorphins are like our natural opiates released in response to pain and exercise. And just in general, drugs and other chemicals affect brain chemistry at the synapses. Okay. Thanks for listening today. That is all we have for module number 9. And have a wonderful day. Take care.